And I wanted to take the first couple of slides just to introduce myself and talk a little bit about my background, which some of this was already mentioned. My bachelor's degree was at Valparaiso University. This is about an hour south of Chicago in the United States. And I had a double major in biology and chemistry and spent some time working in Germany at the time in a Max Planck Institute for organic chemistry. After that, I worked for a couple of years as a research technologist at Rush University Medical Center in Chicago, where I was exploring different joint replacement materials and the response of living bovine and human tissue to those specific components. Then I decided to pursue my PhD at the University of Illinois at Chicago, and the program that I studied was molecular, cellular, and developmental biology. I focused my research on plant biology. So a lot of my research was in the model plant organism, Arabidopsis, where I was trying to understand specific genes and molecular signaling pathways that were used in response to different abiotic stressors, so non-living stresses in the environment. And I also did some applied work at this time with soybean crops and genetic modification of soybeans. After about 5.5 years, I graduated with my PhD, and then I worked for 3 years as a foreign visiting professor at Tecnológico de Monterrey in Guadalajara, Mexico. And so here I met some amazing colleagues like Dr. Maria Roca Sanchez and Dr. Clara Rios Ibarra. And I taught and did research in the departments of medicine as well as biotechnology. Since 2017, I've been working as an assistant professor of biology back at Valparaiso University, where I obtained my bachelor's degree in the United States. And so before I jump into the research, I just wanted to make a little note here that research at the university that I'm at is a primarily undergraduate institution. So all of my work is done with undergraduate researchers, and we don't have any graduate programs, master's or PhD programs. So all the students are undergraduate students that are doing this research with me. And most of them are focused on biology or chemistry, and some of them with goals of going to graduate school and medical school. So today I'll be talking about exploring medicinal plants for novel antimicrobial and anti-cancer compounds. As an example, I'll mainly be focusing on some work in a native Mexican plant that's called Argemone mexicana. I actually learned about this plant when I was working at Tecnológico de Monterrey. Its common name is Mexican prickly poppy or chicolote. And this work was published with 3 undergraduate co-authors, which you can see pictured here on the left, as well as 2 collaborators, Dr. Clara Rios Ibarra and Jeff Pruitt, in April of 2021, so just this year, a couple of months ago, in the PLOS One journal. So largely what I'll be talking about is published work that's in this paper. And so if you're interested in learning more, I'm providing you all the information here so you can take a look at that. And then I'll finish the session by talking about the expansion of this work to other plant species and microorganisms of interest that's been made possible through a NASA grant that I was recently awarded. So my work focuses on finding new compounds and natural drug sources to treat infection and cancer. And the reason I chose to focus on infection or infectious diseases and cancer specifically is because infectious diseases and cancers account for 4 of the top 10 global causes of death, according to the World Health Organization from 2016 data. If you look at low-income countries, the frequency of communicable diseases increases to 7 of the top 10 leading causes of death. And then if you focus on high-income countries, cancer becomes overrepresented. So these are really important illnesses that are present in the global population. Antimicrobial and anti-cancer drug discovery surged in the mid-20th century, but has sharply declined. So this problem is exemplified with antibiotic medications, where 9 new classes of drugs were deployed between 1940 and 1960. But if you look between 1970 and 2005, only 2 unique classes of antibiotics were released. And so this figure that you're looking at from Nature Journal of Chemical Biology also points out a really disturbing trend of antibiotic resistance and shows how quickly superbugs, so microorganisms that are resistant to more than one antibiotic, how quickly they can be created. So this really highlights the usefulness of having many different classes of drugs available and many different sources to obtain those drugs. for the treatment of various infectious diseases. Medicinal plants provide a cheap and easily available source of antimicrobial and anti-cancer drugs. These plants produce a robust supply of novel metabolic compounds. And here, first from 1981 to 2010, about 50% of all cancer drugs originated from natural products. And plants also produce many antimicrobial agents. So here I'm showing you some of the secondary metabolites that plants produce as natural defense compounds, which can act as antimicrobial agents. But with the advent of modern antibiotic drugs that are mainly of synthetic and fungal origins, many plant-derived natural antibiotic sources have been left unexplored. So this kind of sets the context for the research that I'm performing. And I've specifically been looking at Argemone mexicana by its Latin name, which is commonly known as the Mexican prickly poppy. This plant is a very hardy pioneer plant. So it's one of the first plants that can colonize a new environment that's been disturbed either through natural occurrences like fire or through human-made occurrences like industrial destruction of land or agriculture. These are one of the first plants that can start growing in those types of scenarios. They're very resistant to stress and many adverse conditions. And they were native, so they originated in Mexico and the West Indies, but they've now been introduced throughout the world, both by accident, so on the sole of someone's shoe, as well as on purpose. Many people use this as an ornamental plant. Argemone mexicana has been used in traditional medicine for centuries with many reported medicinal properties. This article that I'm referencing here is a review article talking about all the different properties that people have done some preliminary research on in this plant. So antibacterial, antifungal, antiviral, anti-helminth, antioxidant, cytotoxic, and anti-cancer actions. So due to this strong medicinal potential, this plant has been preliminary analyzed to determine some of its main secondary metabolites and compounds present. And some studies have been conducted to better understand some of these activities. But the chemicals possessing many of these biological activities have not been identified, and a few bioactive compounds have been ID'd in the plant. So this is a great plant for a drug discovery experiment. And the goals of this research were to comprehensively evaluate the cytotoxic effect, so specifically antimicrobial and anti-cancer activities of the plants, and to identify the main compounds responsible for those activities. The workflow for this project was to take whole plants and then to separate them into leaves, seeds, inner and outer roots, and then to dry out those plant samples and take 2 grams of each dried sample and homogenize it in specific solvents. So the solvents that were used, I'll talk about on the next slide, but it's used in a 1:4 ratio and allowed to macerate or mix at 200 RPM at 35 degrees Celsius for 3 days. These solutions are then centrifuged to get rid of all large cell debris. And then the supernatant filtered so that only things that are smaller than 0.2 microns or micrometers are remaining, so that small chemicals are remaining in the mixture. And then this filtrate is dehydrated, quantified so that we can normalize the concentration for testing and tested for their bioactivity. The solvents that we chose to use were based on polarity. So we chose 2 commonly used solvents for these types of experiments. One is methanol, that's our more polar solvent, which you can see on the left, that pulls out more polar compounds from the plants. And hexane is a more non-polar solvent, which you can see pictured on the right, and this pulls out more non-polar compounds from the plant. And we tested these purified extracts against 7 unique species of cells. We chose 2 gram-positive bacterial lines, S. aureus and B. cereus, 2 gram-negative bacterial lines, which are E. coli and P. mirabilis. And so those were the prokaryotic organisms that we chose to test our extracts against. We also tested them against the eukaryotic fungal lines, specifically yeast, C. albicans, and S. cerevisiae, and 2 human colon cancer cell lines, T84 and RKO colon cancer cells. So first I'll show you our antimicrobial results. And for these tests, we used an assay called the disc diffusion or Curry-Bauer assay, where the microorganism of interest is swabbed across an entire nutrient media plate. And then a specific amount of the extract of interest, or a positive or negative control, is added to a sterile filter paper disc. And these discs are placed on the plate, and it's all allowed to grow or incubate for 48 hours at the appropriate temperature. Then we can measure a zone of inhibition, so an area where the microorganism isn't able to grow with a ruler in millimeters. And so those are the results you'll be looking at on the next slide. These are the results where 1 milligram of either methanol or hexane extract from either the leaves, seed, inner, or outer root, and you can see the key on the right here based on color, was plated against 6 unique microorganisms. So there's 2 gram positive, 2 gram negative bacteria, and then the 2 fungal lines on the right. The mean zone of inhibition is shown in millimeters for at least 5 biological replicates with its standard deviation. And we always use positive controls, so antibiotics for our bacterial lines or antifungals for our fungal lines. And the negative control was always the solvent alone. So the gram-positive bacteria, as you can see, there's 2 on the left are gram-positive. They were more sensitive to our samples, while the gram-negative and the fungal lines were less affected. And overall, if you take a look at what these purple bars are, we found that our outer root methanol had the strongest effect on all the microorganisms we tested, most of them. And so especially so on those gram-positive organisms. So after this general screen, we started to hone in and focus a little bit more on our root methanol extract for characterization. And we also evaluated the role the developmental state might have on the production of these antibacterial compounds in the root specifically. So for these experiments, we took mature versus immature roots and examined them for inhibitory activity against our S. aureus bacteria. We used 2 grams of either immature or mature roots, and then it did a methanol extraction. And the top panel here is looking at 1 milligram that was plated. The bottom panel is looking at 3 milligrams. And so what we found is that the methanol extracts from the mature root were found to have greater antibacterial activity. So if you look at this row on the bottom and compared to the one right above it, this is the same amount of extract. And you can see that the mature root produces more of these antibacterial compounds compared to the immature root. And so this suggests that these compounds are produced in greater amounts after the plant has transitioned from a vegetative to a reproductive mode. In addition to the antimicrobial activity, this plant has also been reported to have cytotoxic effect against cancer cells, various human cancer cell lines. But colon cancer cells hadn't specifically been tested. And since colon cancer is reported to be one of the leading causes of cancer-related deaths, we tested these extracts against T84 human colon cancer cells for the first time in the literature. And for these experiments, the cells were treated with 1 milligram of our extracts for 1 hour. And then we used an assay called the MTT viability assay that's used to determine cell metabolic activity after treatment with the extracts to determine living versus dead cells. And what you see pictured is the mean percentage of viable cells normalized to the solvent alone for 3 biological replicates. And so what we're going to focus on here are these low bars, the purple and the green. These are the specific extracts that had the strongest inhibitory effect against the colon cancer cells. And once again, our outer root methanol extract pops up, as well as the seed hexane extract. So we chose to focus on that root methanol extract to try to better understand how human colon cancer cells are being affected using quantitative PCR. And this work was done in the lab of Dr. Clara Rios Ibarra. I took some students over to the Tecnológico de Monterrey about 2 years ago, and we worked with her that summer to try to figure some of this out. In these experiments, colon cancer cells were treated with or without our root methanol extract for 24, 48, and 72 hours. And then the RNA was extracted, converted to cDNA, and used for quantitative PCR to figure out the transcript levels of the colon cancer oncogene c-MYC, as well as the tumor suppressor gene APC, which is shown on the right. And the mean transcript for 3 biological replicants is shown here with the standard deviation. And it's also normalized to the housekeeping gene actin. and the transcript level in the negative control is set to 0. So what we found is that the c-Myc RNA levels were found to significantly decrease, indicated by the p-value here of less than 0.05, after 72 hours of treatments. But the APC transcript levels shown on the right were found to increase after every treatment and significantly after 24 and 48-hour treatments. So these results were promising since high levels of c-Myc are associated with different types of cancer, and likewise, since about 70% of sporadic colon cancer is initiated by the inactivation of the APC gene. And so more work is definitely warranted to understand the compounds involved in their mechanisms of action, but these preliminary results indicate that the A. mexicana root methanol extract could possess compounds that modulate different molecular signaling pathways in colon cancer cells. And so next we turned our focus to understanding what bioactive compounds were causing the antimicrobial effects in the roots and leaf methanol extracts. To do this, we used column chromatography to separate individual compounds from our mixtures. And this was based on how they interact with a stationary and mobile phase on the column. Our stationary phase was a silica gel that was used as the absorbance, and then different solvents were run through after we loaded our samples or our extracts that allowed compounds with different polarities to move through the column at different rates. And so that's how we ended up separating individual compounds out of a mixture of compounds. And so the next slide is going to show the results that we got from the roots and leaf column chromatography after plating those eluted fractions against S. aureus. And for panel A, based on the order of the column elution, the methanol fractions were labeled arbitrarily A through F, just in the order that they came out of the column. And then for panel B, the leaf methanol fractions were arbitrarily labeled 1 through 10, based on the order they came out of the column. And some of these we did further fractionation. So the ones that we saw had activity, we did subfractionations, which is why 7 is labeled as 7.3 and 10 is labeled as 10.5. So we did multiple fractionations, subfractionations, and then tested them for antimicrobial activity to figure out which purified compound is having the antimicrobial effect. And the fractions that we found with the strongest activity are the ones that we labeled root fraction D and E, as well as leaf fractions 7 and 10. And so we then evaluated these fractions for purity on TLC plates or thin layer chromatography, and we used them for chemical characterization to try to figure out what compounds were present in these fractions. And so what we did to figure out what compounds were present is we used mass spectroscopy, and this was in collaboration with Dr. Jeff Pruitt in the Organic Chemistry Department at Valparaiso University. This measures a mass-to-charge ratio of ions where a small amount of sample is ionized, usually to a cation by the loss of an electron. And then those ions are sorted, separated, and displayed on a chart. So this is just a general overview of how mass spec works, but it helps us to determine the mass and the number of carbons of the compounds. And so here are our results. Using high-resolution mass spectroscopy, we were able to determine the mass and a predicted structure, chemical formula of our root D and E, as well as our leaf 10.5 fraction. And then we compared this to a list of all known compounds in A. mexicana, and we found matches. Chlorethrine was a compound that was consistent with root fraction D, and berberine was consistent with E, root E, and leaf 10.5. And so we purchased pure chlorethrine and berberine and found that they indeed matched our purified compounds by looking at retention factors, fluorescent properties, and that they displayed similar antimicrobial activity. And so here I'm showing you the structures of chlorethrine and berberine, as well as 2 impurities that still remained in our purified fractions. And as a final confirmation of these matches, these authentic samples of chlorethrine and berberine were used to confirm the identity of the compounds in the roots and leaf subfractions using NMR. And so we've done hydrogen NMR and carbon NMR and confirmed these matches that they're indeed correct. So some conclusions from this work are that we characterize the cytotoxic activities of the seeds, leaves, inner and outer roots from methanol or hexane extracts against 7 unique species and cell lines. And chlorethrine and berberine were identified as the main antibiotic compounds. And we understand a little bit about their distribution now. in the roots and or leaves of the plant. In colon cancer cells, treatment with the A. mexicana roots methanol extract resulted in downregulation of the c-Myc oncogene and upregulation of the tumor suppressor APC gene. And these findings overall highlight the usefulness of plants for the discovery of easily available and cost-effective sources of both antimicrobial and anti-cancer drugs. And so some future goals of this work is to separate other plant extracts and other plant parts of interest in the A. mexicana plant specifically, to quantify levels of berberine and chloerythrine in different tissues of the A. mexicana plant. We're currently working on that right now. To determine a mechanism of cell death in the colon cancer cells, so looking at apoptosis and confirming if that's what's happening in those cells, and trying to identify responsible compounds for the cell death of those cancer cells in the root methanol extracts. And then as pictured on the right here, we're also trying to use the model plant organism, Arabidopsis thaliana, to screen different ecotypes of that plant with different genetic backgrounds in order to identify antimicrobial genes that haven't yet been identified in this model plant organism. And then I was recently awarded funding from NASA through an Indiana space grant to expand this work to test specific organisms that have been identified on the International Space Station that have resulted in superbugs on the International Space Station, and to expand this research to include more plant species. This grant includes an educational component to incorporate an authentic research experience early on in an undergraduate student's career. So we're going to include research relating to this in both the introductory microbiology labs at Valparaiso University and in the organic chemistry labs at Valpo. So with that, so I have time for questions at the end. I'd like to thank the organizers of the Bolivia Innova for asking me to give a presentation. I'd also like to acknowledge my undergraduate researchers. So these three researchers helped out a lot with this work. Teodora Nadeska, who's on to graduate school, TJ Lefebvre, who's going to dental school, and Stephanie Bocangel Gamara, who also recently graduated and is moving on to work in a vaccine lab, development lab. I'd like to acknowledge my collaborators, Dr. Clara Rios Ibarra, as well as Dr. Jeffrey Pruitt, for their work for Dr. Clara on specifically on the qPCR work with the colon cancer cells, and Dr. Pruitt's in the organic chemistry lab for the mass spec and the NMR, nuclear magnetic resonance. And then this funding was made possible through internal startup funds and through a Wheat Ridge grant from Valparaiso University, as well as through external funding agencies, which are the American Society of Plant Biologists and the recent NASA Indiana Space Grant as well. So thank you. I'll show you my references. Thanks a lot for attending this talk, and I'd be happy to take any questions you have at this time.
Oh, thank you very much, Danielle. I find it fascinating that you do all this research without the need of PhD students. I think that's very interesting. I'm going to read a few questions that we have. First, Maria Mercedes Roca, that I understand you know, asks, what effect do you think these plant antimicrobials would have on the gut microbiome? Do you think we should be concerned about damaging our microbiome in an attempt to inhibit pathogenic bacteria?
Yeah, I mean, that's a great question. We haven't specifically explored that, but that's an excellent question, and definitely something that could be evaluated to look at some of the toxicity effects. In this case, we're more isolating individual compounds for, antimicrobial or anti-cancer treatments. But if you were using it more in like a traditional medicine sense, maybe you would ingest the entire plant, and that definitely be something to take into consideration. Yeah, that's a great question.
Okay, thank you. Kenji Shoji, who is also a member of the organizing committee, has two questions. I'm going to read both of them. Thank you for your presentation, very nice work. Have you tested your compounds in normal colon cells? Do you think the effect on cancer cells is specific to malignant cells? Do you have any clues of the target for these compounds? And the other question is, are you going to test these compounds in other cancer cell types?
Yeah, so I'll start with the first question. I think I'm going to break that down into three questions. So the first one, was asking about, can you repeat the first question? Sorry,
Yes.
already lost
Have you tested your compounds in normal colon cells?
Oh yeah. Okay, so we would like to do that, but have not yet. We have looked at eukaryotic lines, different fungal lines, but as far as the human cell lines, we haven't looked at normal colon cancer cells, and we definitely would like to do that. We did, as I showed, looked at some of the changes that are happening in these colon cancer cells in regards to the transcript of specific oncogenes and tumor suppressor genes. So that does give us a little bit of information about the mechanism of how it might be acting, although a lot more research is warranted to really understand that. But that is definitely something we'd like to explore. And then what was the second part of that first question?
Yeah, do you think the effect on cancer cells is specific to malignant cells?
Oh, yeah, so I think that still relates to the first question there, that we should definitely look at that and see what differences there might be for those assays for non-cancerous colon cells.
Well, I think you also answered, do you have any clues of the target for these compounds?
Oh, yeah. So actually, for the specific, if you remember back to that very first antimicrobial, just the general screen that I showed you, how they were targeting more the gram-positive bacterial cell lines. And so we had some clues at that time that we thought it might be something targeting the peptidoglycan cell wall for its antimicrobial activity, because that's one of the major differences between the gram-positive and gram-negative bacteria. And then one of those compounds that we identified, I think it was chlorethrine, does specifically target the cell wall and is present in the roots of those plants. So that confirms our idea that one of the targets, at least for the bacteria that it's targeting, that's not related to the cancer cells, but one of the targets for the bacteria is the peptidoglycan cell wall.
Okay. Yeah, and the last question, I think you already answered if you're going to test these compounds in other cancer cell types.
Yeah, and so we'd like to explore some more cell types that haven't been explored yet. There is some research, more just looking more like at the MTT assay in different cancer cell lines, not so much at the like looking at transcript levels or qPCR. So those are some questions that could definitely be explored in different cell types that hasn't been published yet in the literature. It's a great idea.
Okay, thanks. I have another question from Claudia Figueroa. She asks, how is the anti-cancer potential of an organism, plants, mushrooms, etc., recognized at the initial stage of investigation? And I also had sort of a similar question. You presented the results of one plant, but I am very curious about the screening. I don't know if you have some ethnobotany stage before all this lab research.
Yeah, the reason I actually focus on this plant, someone in the university that I worked at in Mexico had told me about this plant and knew that I had a plant biology background. So that's the reason I started looking at this plant. And then I did a literature review, saw that literature was lacking as far as to understanding specific bioactive compounds. And that's kind of what put me in this direction. But there are a lot more, interesting questions and interesting plants. And we do hope to explore some more explore this more broadly with that grant that I talked about at the end that has that educational component, and looking at this in the classroom and having the students do work on different medicinal plants using similar screening methods to what I presented here.
Okay, we have one more question from Natalia Montellano. She asks, did you try different solvents at the beginning of the research? How did you decide the mix methanol benzene?
Yeah, we did. We tried lots of different solvents, maybe 5 or 6. I tried to use things that were common in the literature that's obviously that were used for these types of experiments and that had a different range of polarities. And so we tried some that we just didn't see much of an effect with, like water, for example. We used aqueous solution as our solvent, just using straight water. Some issues with that are that it took a really, really long time to evaporate, and that we just didn't see a large effect. So we did look at different solvents and then tried to narrow in where we were seeing an effect, and then what was feasible to actually work with in the lab as well for the type of lab setup that we have.